You're listening to Conferences Online Allergy from Children's Mercy Hospital in Kansas City, Missouri. Today is November 6, 2023. Our topic is How the Immune System Works by Dr. Lauren Sompayrac, chapters 8 through 16. This is presented by Dr. Nikita Raje. She is an associate professor of internal medicine and pediatrics and is as chief in the section of allergy and immunology at Children's Mercy Hospital.
So here are the learning objectives. These are for chapters 8 through 12. I did add 13 to 16 chapters as well. And some of the chapter numbers were reshuffled. So vaccines is actually chapter 14. But we'll be talking about all these chapters, and here are those learning objectives. Let's start with restraining the immune system. So last time we talked about how there is activation, and once there is activation, the cells differentiate into the effector molecules so that they can actually provide the immune response. So once that's completed and the immune response is successful, it's important that the immune system is not active for a long time. So there are several ways the immune system can be controlled and the immune response is waned down. So let's look at some of those mechanisms. So how is the immune system attenuated? So there are cells called inducible Treg cells. So last time you heard about T helper subsets that can be coached to become different types of subsets: Th1, Th2, Th17. And similarly, there are some other T helper cells that can actually be induced or coached to become Treg cells. These are called inducible Treg cells. So for an example, let's think about intestine, where there is TGF beta. And when the TGF beta is in abundance in the environment, the dendritic cells coach the T helper cells to become iTregs or inducible Tregs. These inducible Tregs then produce TGF beta and IL-10. So these are anti-inflammatory cytokines. So instead of producing, say, IL-17 or IL-12, interferon gamma, all of those are inflammatory cytokines that help to provide the immune response against the microbes. But then these are the cells that, if these are activated, T helper cells that are induced to become Tregs, when they are activated, they produce anti-inflammatory cytokines, TGF beta and IL-10. And so these cytokines then make sure that the rest of the immune system actually becomes less active or is deactivated. So how do these cytokines work? So TGF beta reduces the proliferation of T cells, and they make CTL, cytotoxic T cells, less vicious killers. IL-10 also helps in blocking the co-stimulatory signals, so it becomes difficult to activate naive T cells at that point. So when the immune system's actually done its job, you want these cytokines around so that they can calm down the immune system. Oh, all right. So so the same thing, but there are several mechanisms in place of how that immune system is deactivated. So for one, once the immune system controls the infectious particles reduce, the invaders are destroyed. So whether it's a virus or bacteria, the number of viruses or bacteria that are alive to activate the immune system goes down. So there is less antigen present. So there is less activation of the innate immune system, such as antigen presenting cells. So there is less activation of the adaptive immune system, because the adaptive immune system needs the innate immune system to activate it. So overall, there is deactivation of the immune system. Also, the immune mechanisms that are already present are going to be have short life. So basically, neutrophils that were activated or called upon as first responders, they only live for a few days. Natural killer cells that are activated live only for a week. Macrophages, while they live for a longer time, if they do not get the activation from interferon gamma, they'll go back to their resting state rather than being activated or hyperactivated. The dendritic cells also live in lymph nodes for 1 week. Plasma B cells that produce antibodies are only alive for 5 days. And then the antibodies that are produced by these cells have a short half-life, the maximum being for IgG, which is 3 to 4 weeks, as we know. So overall, all these mechanisms that are in place have a short life. So all the mechanisms that were produced because of the activation are going to be gone in a few days or a week. And then if there is no further activation because that the antigen has gone down, these mechanisms are not good. Those weapons are not going to be produced further. Also, what about the cells that are already activated, right? Or there are some naive T cells that are still present, which may be around to get activated if there is less, even if there is some antigen present, the remaining antigen that's present around. So why does that not cause more problems? And the reason is there are some checkpoint proteins that decommission the T cells that are present. What does that mean? So we know that the antigen presenting cells come in contact with the naive T cells and activate them using some of the co-stimulatory molecules like B71, B72 that will interact with CD28 on the naive T cells. However, when there is enough activation that has already occurred, those experienced experienced T cells, which were activated from being, these are the naive T cells, they become activated, they have gone through their process of helping with the immune response. Those are experienced T cells, then start expressing CTLA4 on their surface. What's the difference between CD28 and CTLA4? They both interact with B71, B72. However, CTLA4 has 1,000 times more affinity for the B71, B72. So when CTLA4 is present, the B71, B72 are going to preferentially bind to CTLA4. And CTLA4 is not present on the naive T cells. So initially, the B71, B72 don't have a choice but to engage with CD28. But once activated T cell has gone through that experience of being around for a while, CTLA4 is going to be expressed, and then B71, B72 will interact with CTLA4, which is deactivating protein. And so because of that, the T cells will not be reactivated. Similar phenomenon occurs with another protein called PD-1. PD-1 is present on activated T cells, and they can interact with its ligand called PD-1 ligand that's present on inflamed cells. So when the cells are inflamed for a while and they have gone through that inflammation process, the PD-1 ligand is going to be expressed that will interact with the activated T cells and prevent the proliferation of activated T cells. So then another concept of calming down the immune system is AICD, or activation-induced cell death. So after there is multiple re-stimulation of T cells, those T cells are eliminated. They can be eliminated by AICD. One such mechanism is FAS and FAS ligand. So FAS ligand is on CTLs, and it can bind to FAS protein. So the naive T cells do not express FAS, and they are insensitive to binding to FAS ligand. But the activated T cells will express FAS protein, and that will interact with FAS ligand and will eventually be asked to undergo apoptosis. All right, so that's how the immune system is restrained once the function is done, right? But we also need to know how there is a system in place so that the immune system is not reacting to self-antigens. That's called self-tolerance or MHC restriction. So let's look at this concept. So there are different layers of tolerance. So the first step is central tolerance. Central tolerance is present in central immune organs, such as thymus, which is a primary lymphoid organ that has the structure including cortex and medulla. There are no incoming lymphatics, but the T cells are delivered to the thymus. The cells that are destined to become T cells will be delivered to the thymus, and but there are no HEVs, there are no high endothelial venules. So where is the antigen coming from? So that's the part that we are going to learn here. So these cells that are destined, the lymphoid progenitors, are delivered to the thymus, and those cells are called nude cells that do not have any markers. They do not have CD3 on them, they do not have CD4 on them, or CD8 on them. Once they go through their cortex and they interact with some cortical thymic cells, they can become double positive T cells. These double positive T cells then basically interact with cortical thymic epithelial cells, where they undergo MHC restriction, which means these double positive T cells will come in contact with either class 1 MHC or class 2 MHC, on the surface of which these thymic epithelial cells will express the self-antigens. They express class 1 MHC, which will present the antigens that are produced in the thymic epithelial cells. However, they also have class 2 MHC, and what cortical thymic epithelial cells do is they undergo a process called autophagy. What does autophagy mean? Autophagy means that when the cells, autophagy can occur in different times, but in cortical thymic epithelial cells, it's for a different reason. What autophagy typically means is if the cell is under stress, that cell can use its lysosomes to make pouches to collect the internal cellular proteins and put them in lysosomes. Then they will be dismantled. So the proteins are dismantled into smaller molecules that can be later reused by that cell once that stress is over, to form the cellular proteins needed for survival. So it's not a dead cell. However, autophagy means all their cells cellular proteins are going to be proteins or other molecules are kind of, put into lysosomes and put in pouches for later use. Cortical epithelial cells will use that mechanism of autophagy to collect the internal proteins into lysosomes, and then those can be expressed on the surface of class 2 MHC. Remember, that's how the class 2 MHC expresses the proteins on its surface, using anything that's collected into that endolysosome. And then that's put on the surface of class 2 MHC and then expressed to the double positive T cells. Once that occurs, these cells become single positive cells. Either class 1 or class 2 MHC is expressed on that surface of that's presented to those cells, so to become CD8 or CD4 cells. These cells move on to the medulla and come across thymic dendritic cells and medullary thymic epithelial cells that present self-antigens, again, on the surface of class 1 and class 2 MHC to these single positive cells, so that the cells that are reacting to self-antigens are killed, and then the ones that are reacting only to, do not react to self-antigen but can recognize MHC, are going to survive into those single positive cells that are going to be released from the thymus. So that's the process of MHC restriction. So same process here, just shown in a different way. The cells that enter into the thymic cortex initially are they are in proliferative phase and they do not undergo apoptosis. But as they move through cortex and medulla, these cells turn into double positive cells that are very prone to apoptosis, and depending on whether they are able to go through positive and negative selection, if they are unable to pass those tests, they will be killed to the point that they start out in the numbers of 60 million, and when they graduate, there are only about 2 million that pass both those tests of positive selection and negative selection. So what's positive selection? The cortical thymic epithelial cells ask these T cells, do you have receptors that recognize one of the self-MHC molecules which I'm expressing? If it can recognize self-MHC, it passes the test. That's the reason it's called positive selection. So the answer is yes, and that's the reason it's called positive selection. The medulla asks a different question to these developing T cells. The question is, do you recognize any of the self-antigens that I display on MHC molecules? And the correct answer is no. If the T cells that are developing say, no, we cannot recognize the self-antigens, then it's going to survive. That's called negative selection. And the thymic graduates, as I mentioned, if they have to pass both these tests to graduate from the thymus and come out of the thymus, this whole process takes about 2 weeks. All right, so that's the first step of tolerance, self-tolerance. However, there are multiple layers of tolerance built into our immune system so that the T cells that are coming out of the thymus, if they are if they have escaped, if the self-reacting T cells have escaped, there are several layers of tolerance built so that they can not cause more problems for the human body. So one such mechanism is tolerance by ignorance, which means, say, a naive T cell comes into the circulation from the thymus, and it can actually react to a self-antigen. But however, these naive T cells recirculate from secondary lymphoid organ, from one lymph node to another, one secondary lymphoid organ to another, but they don't go into the tissues. Naive T cells are supposed to stay in the recirculation pattern till they come across the antigen in this, say, a lymph node, and then they are going to be activated. So then they are no longer going to be a naive T cell at that point. So the naive T cells are only in this recirculation going from one lymph node to another, right? But the self-antigens, typically, that would activate these naive T cells are not going to be in the secondary lymphoid organs. They are going to be in the tissue. Say there is a self-antigen in secondary lymphoid organs that are likely the ones in the thymus, so that those T cells that are reactive to those self-antigens are going to be eliminated in thymus. But the rare self-antigens in the thymus that are not present in the thymus are also going to be rare in the secondary lymphoid organs. So the pattern of the traffic where the naive T cells move from one place to another, there are not enough self-antigens that they can react to. That's called tolerance by ignorance. Another issue is that what if there are some naive T cells that may come across an antigen that it can react, self-antigen that it can react to? Well, to handle those, there are these natural Treg cells that are produced in thymus. These natural Tregs have CD4. These are CD4 positive T cells. They have been come in contact with the medullary thymic epithelial cells that present the self-antigen to them. But instead, if they can react to them mildly, those, instead of being killed, start expressing FOXP3, a protein that turns them into natural Tregs. These can enter the lymph nodes and secondary lymphoid organ and protect against the T cells that can react to the self-antigens. Apart from that, there is peripheral tolerance, which means the naive T cells that are typically present in secondary lymphoid organs. What if it actually goes into the tissue where they can come across the self-antigen that they can bind to? If that happens, thankfully, there are not, because there is no infection, inflammation going on, the antigen presenting cells are not in that activated or hyperactivated state. So they do not express co-stimulatory molecules that are needed to activate these naive T cells. So even if they come across the antigen and they can bind to them, they do not have the second signal, so they are not activated. That process is called energy. Versus if they are in the secondary lymphoid organ, they would actually get activated. Instead, in the peripheral tissues, these naive T cells, even if they come across their antigen, they will not get activated but undergo energy. And then what if the self-reactive naive T cells escape the thymus, enter the tissue, find enough antigen, and there are co-stimulatory molecules? In that case, there is the process called AICD, which means there is activation-induced cell death. So maybe they will cause a little bit of a problem, but eventually those T cells are going to be killed because they have been activated for a long time. Apart from T cells, B cells are also supposed to have tolerance to self-antigens. How does that happen? Now, B cells are produced in the bone marrow, but then they complete their maturation in the periphery. Um, in the during that process of development of or maturation of B cells, there is gene recombination that occurs to form the BCR. If the BCR can bind to a self-antigen in the bone marrow, those B cells are going to get a second chance of editing the receptor. That's called receptor editing. This phenomena is really important because the T cells don't get that second chance. In the bone marrow, the B cells actually get a second chance because if it's a self-reacting T cell, it's going to be killed right away in the bone marrow. But the B cells get a second chance at editing their receptor to see if they can come up with a different combination of gene segments that is not going to react to self-antigen. So now say the B cell is produced, it has a BCR, but unfortunately it escaped that receptor editing and it's a self-reacting B cell. That naive B cell is going to be released into circulation. But again, because it's a naive lymphocyte, it's going to follow a pattern in the traffic where it's going from one lymphoid organ to another. So the rare self-antigen is not going to be come in contact with that naive B cells in the secondary lymphoid organs. That's the same phenomena called tolerance by ignorance. Similarly, naive B cells that enter the tissue, if they come across that self-antigen, they will not have that enough innate immune response so that they undergo energy. That is in form of no T cell help as well, along with the innate immune system. And then the germinal center in the lymph nodes typically produce high affinity antibodies due to a process called hypermutation, somatic hypermutation. During that process, sometimes the B cells can make BCR that was not initially self-reactive, but later on, due to that hypermutation, it can cause to produce a B cell that's self-reacting. In that case, it's going to actually be tested against the follicular dendritic cells that only display the opsonized antigen, which means that the self-reactive B cells are going to be killed and they are not going to undergo further proliferation in the germinal center. So that helps in providing B cell tolerance as well. NK cells also have some tolerance, right? It's important that all parts of the immune system are reacting only to the foreign antigens. So the NK cells have inhibitory receptors that are looking for interaction with class 1 MHC. If that class 1 MHC on a cell is absent, in that case, those NK cells are going to kill that target cell. However, the NK cells that do not recognize self-MHC and class 1, those are rendered non-functional to make sure that the NK cells that are later on released from the bone marrow are not reacting to self-antigens. All right, moving on to the next chapter, immunological memory. Both innate as well as adaptive immune system have some memory. Innate memory is pretty much basic memory, which means it has memory for the broad class of molecular patterns, such as for TLR or NK cells. Um, However, it's trained immunity. It's short-lived. Such as for macrophages, they may have short-lived memory, so that they will look for the same pattern that they've have been looking for at that time of that infection. That memory stays for local area and doesn't get transmitted throughout the body. And that trained immunity is driven by epigenetic modifications. This innate immunity is not adaptable. Versus the adaptive immune system, B cells and T cells have adaptable memory. B cell memory actually needs T cell help. They make long-lived, so here is a naive B cell. It can be activated, and those activated B cells can either become short-lived plasma B cells, which will produce antibodies for the quick response, but they can also become either central memory B cells or long-lived plasma B cells. And those long-lived plasma B cells will stay in the bone marrow and produce small amounts of antibodies. Or the central memory B cells will be present in the secondary lymphoid organs, not the bone marrow, but the secondary lymphoid organs, where they will replenish the long-lived plasma B cells over time. And they can also, when needed, turn into short-lived plasma B cells to produce antibodies in case of repeat infection. T cell memory, again, a little different. Naive T cells get activated. Activated are differentiated into effector T cells, and those effector T cells can form effector memory T cells, right? Effector memory T cells, say these effector T cells are in liver. These effector memory T cells will also be present in that tissue. However, the central memory T cells will be present in secondary lymphoid organs, and they can actually, if again there is another infection, they can turn into effector T cells. Also, there are some, sorry, did I say? So those are the tissue resident memory T cells that stay in the tissue. And then the effector memory T cells are going to circulate in the blood or lymph. So secondary lymphoid organs, tissue, and in blood and lymph. So three types of places where the memory T cells reside. Immunological memory has specific properties for the adaptive memory. So we know that 1 in million cell, right, in among all the naive T cells is specific for a particular antigen. When that infection occurs, the infection is cleared, and most of the T cells that were activated are going to be killed. However, there are few memory cells that stay back after the infection. Those memory T cells are still higher in number. They are very few, but they are higher in number than when they started out as naive T cells for that particular antigen. Again, the memory T cells are easier to activate. They need about 50 times less MHC peptide for their activation, and really they don't need co-stimulatory molecules at that point. And the B cell memory has even more features. They have an upgraded B cell memory in form of class switch memory, which means the B cells got activated, they formed IgM, then they switched to making IgG, or in case of parasitic infection, they started making IgE. And if there is a repeat infection, the memory B cells that were produced during that infection, instead of going from IgM and then switching to G or E, they will directly start producing that particular class of immunoglobulins. Also, the affinity affinity of the antibodies that's produced from those memory B cells that underwent somatic hypermutation and had affinity maturation will produce long-lived plasma cells that are gonna produce antibodies directly of higher affinity in case of repeat infection. All right, so that's what is immunological memory. There's a difference between innate and adaptive memory, as we talked about. Innate memory is not updatable and is same for all the humans, versus adaptive memory is expandable, and actually starts out at zero. Even the naive B cells and T cells are present in a baby, they may not have any memory initially, and then it's pretty expandable at a very quick rate, and it's personal. It's different for each individual. Moving on to intestinal immune system. Um, we know that the intestinal immune system has specific features that are specific for the intestine. So we know that there is a physical barrier that's provided by epithelium, and then there is mucus that's produced that causes a formation of mucosal layer on top of that. The mucus provides a barrier, physical barrier, along with the epithelial layer, but it also has proteins that have antibacterial properties. Those are called lysozymes and defensins. Defensins can be alpha defensins and beta defensins. And the mucus helps, that layer helps to sweep out all the bacteria in the lumen. The bacteria that are present in the intestine also play a pretty important role. All of them together, bacteria, viruses, parasites, all of those that live in the intestine form the intestinal microbiota. Among them, there are bacteria that are called commensals, which means they live in the intestine, and there is a symbiotic relationship where they help the intestine as well as they get their nutrition from the intestine as well. The commensals can break down carbohydrates, make vitamins, and they compete with pathogens so that the pathogens don't get the means of survival. But we know that there are times when there can be intestinal invasion. And so the tricky part is that the immune system has to handle the intestinal bacteria very gently, because there may be commensals that they have to interact with, but they cannot ignore them, because if they continue to ignore them, those commensals can then actually enter the circulation and cause infection. So they have to handle them gently. So how does that happen? Typically, it has a non-inflammatory cytokine milieu, because of which there are going to be cytokines like TGF-beta that's present around, so that there is no inflammatory response in the intestine. But at times there can be inflammation, because if there is a problem with some of the bacteria or some of the microbes, there are resident macrophages, dendritic cells that will react to those particular antigens. Those antigens are going to be carried to the draining lymph nodes and will activate the T and B cells there, and they'll also attract neutrophils that will give rise to, all of them together give rise to inflammation. The antibodies that are produced by the B cells in the lamina propria are more kind of adapt to switch to IgA production, even without help from T helper cells. So remember, for class switching from IgM to IgA, typically B cells need help from helper T cells. However, in case of intestine in the lamina propria, the B cells are adapt to become to class switch to IgA. That's driven by retinoic acid. The retinoic acid is produced by intestinal dendritic cells, and that retinoic acid works on the B cells to class switch to IgA. The IgA that's produced is really important. It stays from the lamina propria. It can undergo transcytosis. It can bind to the luminal bacteria and bind those bacteria so that it's actually either swept away or the bacteria are kind of neutralized because they cannot breach the barrier in that case. However, that's IgA that's going to bind to it, but not cause inflammation, which is important for the intestine. Some of the features of intestinal immune system are really important to know. That there is the distributed response, which means if the B cells or T cells are activated in one part of the intestine, It's not only at the site of the breach where the bacteria tried to breach the barrier. That at that time, those B and T cells are going to be can move along the surface of the intestine so that the bacteria at other places cannot break that barrier. However, there is a private response, which means even though it's distributed throughout the intestine, those activated T and B cells, they are only in the intestinal lumina or lamina propria, not in the systemic circulation. And we already talked about this. There is an anti-inflammatory environment with the iTregs giving rise to TGF-beta and IL-10 anti-inflammatory cytokines. That's really important. How do the common cells help in the anti-inflammatory action? So we know that the common cells produce some molecules or some substances like butyrate that helps in activating the helper T cells to become inducible Tregs. There is a substance that B. fragilis produces, polysaccharide A, that will induce the T cells to produce IL-10. And then Bifidobacterium breve that induces the dendritic cells to produce IL-10. So all anti-inflammatory actions. What if there is actually a pathogen that infects the intestine? In those cases, so remember, at rest, there is an anti-inflammatory environment with TGF-beta. However, and so in that case, there will be iTregs that are produced. But when there is an infection, the cytokine that's produced at that time is TGF-beta along with IL-6. And in presence of that, there are T cells that will be induced to become T helper 17, Th17 cells, or Th1 cells in presence of IL-12. And so the Th1 will produce interferon gamma and class switch to IgG, and Th17 will produce IL-17 and attract neutrophils to the site. And so that will give rise to more inflammation that's needed to fight that infection. All right, next chapter, immune system gone wrong. Immune system can go wrong and cause allergies. It can cause autoimmunity. It can cause various features, various different types of autoimmune diseases, which could be due to self-reactive B cells or self-reactive T cells. So that's just a slide showing what could happen at that point. Next one is vaccines. We know there are different types of vaccines that we use. Those are killed vaccines, which means those vaccines use substances like formaldehyde to kill the whole virus and use it as a vaccine, like polio or influenza. There are some toxoids, such as diphtheria and tetanus bacteria that produce toxoid. That toxoid is purified, treated with aluminum salts, and then used as a vaccine. And then there are acellular vaccines, which means the cell is used, like the part of the vaccine microbe is used as a vaccine, such as pertussis. Subunit, which means viral proteins are produced by genetic engineering, like hepatitis B or HPV. And carrier vaccine, where a gene is introduced in a non-disease causing virus and used as a Trojan horse. And then RNA vaccines, such as in COVID-19. All right, the attenuated vaccines are a little different. Those are actually live vaccines, such as MMR and varicella. The big difference between these two is the non-infectious vaccines will induce memory helper response and B cell response, whereas the attenuated vaccines, because those are live vaccines, will create memory helper response, B cell response, as well as CTL response, and that CTL response is a really important part of that response to produce memory of these microbes. And so a couple of other features of attenuated vaccines is that it can provide herd immunity. It can also cause a problem because it can lead to infection in immunosuppressed individuals. But overall, and And typically it doesn't, but yeah, mutation can occur in that virus that can cause disease in immunocompetent host. Vaccine adjuvants are used in some of the vaccines that we talked about. Why is it used? Because the antigen itself helps to know that it's a foreign antigen, but the danger signal is provided by the adjuvant in case it's not a whole killed vaccine, right? So if it's part of the microbe, then it will not give that danger signal. And in those cases, adjuvants are important. Couple of them that are used very commonly are aluminum hydroxide or LM and modified LPS. All right, moving on to chapter 13, it talks about immunodeficiency. Not going to go into details of these, but we know that there are different types of immunodeficiencies or inborn errors of immune system, such as a combined immunodeficiency syndrome like hyper IgM syndrome. We talked about how CD40 ligand are important for interaction between T and B cells. If that's defective, the communication is lost and it causes hyper IgM syndrome. Complete absence of B and T cells can lead to severe combined immunodeficiency. Complement proteins can be defective, such as C3 deficiency. Neutrophils can have problems, such as defect in adhesion molecules can lead to leukocyte adhesion defect. Absence of mature B cells will lead to absence of antibodies, causing A-gamma globulinemia. Treg defects can lead to immune dysregulatory disorders. Innate immune defects can be present in presence of TLR defects. There can be autoinflammatory syndromes, such as defect in IL-1 receptor. And then there are bone marrow failure defects that lead to complete absence of some of the cell lines. So these are different types of inborn errors of immunity. And then, of course, these are all mostly congenital. They are all primary immunodeficiencies versus there are secondary immunodeficiencies. One such big example is HIV/AIDS. And this figure basically shows how there is acute infection when the virus particles are going up. At that time, there are enough CD4 cells and CD8 cells, but the virus-specific CTLs are pretty low. Over time, as the virus-specific CTLs go up, the viral particles go down, and then the helper T cells over time continue to go down. CD4 cells are the ones that they infect. So over time, they go into that chronic phase and then profound immunosuppression unless there is antiretroviral therapy that's used. All right, moving on to the next chapter about cancer and the immune system. So remember, cancer is caused because there is an imbalance between two types of genes, proto-oncogenes and tumor suppressor genes. What's the difference between proto-oncogenes and oncogenes? 1 second here, let me realize that my chart got displaced. Right. So proto-oncogenes are the genes that if mutated will turn into oncogenes. If there is presence of oncogenes, those oncogenes will cause cancer. Tumor suppressor genes like P53, those help to suppress the proto-oncogenes. If there is an imbalance between those two, it can result in cancer. Cancers can be of different types, or malignancies can be of different types, such as carcinomas and sarcomas. Blood cancers can be leukemias or lymphomas. These cancers can be spontaneous or be induced by viruses. And so there are different parts of the immune system that help to perform surveillance against cancer cells. Most important being cytotoxic cells, CTLs, but also NK cells and macrophages. All right, then we talk about immunotherapy, which is provided by both B cells and T cells. These are produced by humans to tackle some of the immune issues that we come across, such as hybridomas. Those are produced. Hybridomas are produced when you cross. Can you see the slide? What just happened? Okay. Um, when the plasma B cells that make one specific type of antibody is fused with an immortal cancerous B cell line, both of those cells can fuse to form an immortal antibody producing B cells called hybridomas. Also, there are some cells that are used to treat cancers called CAR T cells. So um, so what are these CAR T cells? These are actually a combination of three different parts. And that's based on actually a Greek mythological figure that has a head of a lion, body of a goat. Um, I forget what the third part is. Tail of a dragon. So same way, CAR T cells has three different parts. The part that binds to the antigen, tumor antigen, is a single chain antibody. So it has a heavy chain and a light chain. Instead of a full antibody that has two heavy chains and two light chains, it has one heavy chain, one light chain that can bind to the tumor antigen. That is fused with a signaling chain, CD3 zeta chain. And then that is also fused with a CD28, which provides co-stimulation. So we learned in the previous chapters that T cell activation needs a TCR, T cell receptor that can bind to the antigen that's provided by the external single chain antibody here. Then that signaling has to go through the CD3 chains. So CD3 zeta chain is attached to it. And then it has to have that second signal provided by CD28. And that is also included in this CAR T cell. So CAR T cells are used they are engineered so that they can bind to certain tumor antigens, and those engineered T cells are given to a patient with cancer so that these CAR T cells can interact with those tumor antigens and kill the cancer cells. All right, so that's the end of